Good morning everyone and welcome back to another video. My name is Jane and I'm a second year PhD candidate in cancer research. So today is Tuesday. It's the first day of my working week because yesterday, Monday, was a public holiday for the Queen's birthday. Thank you, Queen. So it's 12 p.m. currently. I ended up having a really busy morning and I arrived a little bit late. So I went straight into the lab and was checking my cells and things. So all of my cell lines are looking healthy and happy and not contaminated, which is really good news. So today I'm going to set up another colony assay after I took out my colony assay last week. I'm going to see less cells than I did last time, so 1,000 cells per well, and see how that goes. I'm also sitting down today and planning a bigger time course for either next week or the following week, which is going to be a massive day in the lab and requires a lot of preparation and a lot of planning. So that's about it from me this morning. Wish me luck. So it's 5:45 p.m. I started my cell culture at 2 p.m. So I have been in cell culture for nearly 4 hours now, with the occasional break, usually just like a sip of water or a snack. I am exhausted.
Welcome to Jurassic Park. They're moving in herds.
Hi everyone. Today is Wednesday. It's currently about 10:15 in the morning. I am at my university campus because I attended a counsellor session this morning. I am a firm believer that mental health is of the utmost importance, and PhD students often suffer from mental health issues. So I try to keep on top of mine by attending counsellor appointments regularly to talk about the things that are bothering me and blah blah blah. So if you're watching this, to my counsellor, thank you so much for your support. You're amazing. So I'm about to catch the bus back to the lab. My lab is at a different campus to my university campus, but it's close by, which is handy. So I'm gonna go and check the colony assays that I set up yesterday and see what is happening in the lab. It's now 3:45 p.m. I am currently in the lab making up some SDS page gels for tomorrow, so I will run some gels and do some more protein analysis. I do a lot of protein analysis. I'm also doing some planning, so I'm planning some pretty big experiments over the next 2 to 3 to 4-ish weeks, so yeah, should be exciting and busy. Good morning. Today is Thursday. I am really glad that I made my SDS page gels yesterday, because today I have a massive day in the lab. I need to measure my protein concentration from some protein samples that I extracted on Monday, I think. I need to make up those samples and then run those samples on the gel. All up, it's probably going to take me about 7 hours. I'm hoping I can make it to liquid nitrogen at some point today too. Maybe. I'll see if I get time, because I also have some cell culture to do today. Wish me luck. It's a big day. So the thing about working with protein is that it needs to be kept cold, or else it will be degraded. So the first thing I need to do is grab an esky and get some ice out of our ice machine. So an esky is basically an Australian word for an ice bucket. Good morning, everyone. Today is Friday. I'm glad that it's Friday, but at the same time, I will have to pop into the lab this Sunday to do some cell culture, so I'm not too excited for the weekend, which kind of sucks, and I think I'll have to do some work next weekend as well, but this is just a part of doing a PhD. Sometimes you cannot help but do some work on the weekend, which is fine. So yesterday I ran my gels, and They were all looking good after their transfer last night, so I'm just about to go in and get my membranes and start the washing process and get some results in a few hours. So I hope you all enjoyed my video this week. I finally got to go to take my cells down to liquid nitrogen, but unfortunately when I opened the box where I thought my cells were being stored, the box was empty, so I will have to try and find those cells at some point when I get time over the coming weeks. It's possible that the lids have just been mixed up in the liquid nitrogen, but I'm not 100% sure where they are. So far my colony assay is going well. Hopefully we should have some good results from that in the next week or so. And my protein results on Friday were not what I was expecting them to be, so I may have to do some troubleshooting in the coming weeks. If I get time, I have a really busy couple of weeks coming up, so we'll see how that goes. So if you liked this video, please give it a thumbs up, and if you would like to see more videos like this, please hit the subscribe button. And if you would like to see what happens when someone walks into the lab while I'm being weird and filming on my phone, please continue watching. There's a funny clip at the end of this video. Otherwise, happy sciencing!